Right into your presentation. Thank you so much for coming. And so today is my honor to present or to introduce our last, but not least, of course, one of the best neuroscientists, Mehrdad Jazayeri, who is an associate professor right now at MIT. And apparently he is a Sharif University graduate. So I have to say welcome home, because this is a Sharif symposium. It's good to have you. Mehrdad has done his master's degree in physiology in University of Toronto and PhD in NYU. He has worked with Tony Mawshan. He has done postdoc with Mike Shadlin, and he has done a beautiful work in time perception and decision making, and other recent work in hierarchical cortical and like a brain computations. He is awarded the Helen Hay Whitney Foundation Fellowship for postdoc. He is the recipient of Alfred P. Sloan Research Fellowship and McKnight Scholar Award and other awards, and very good publications. And currently his lab studies how dynamic patterns of neural activity in the brain give rise to algorithms that support mental computations. And so we cannot wait but to hear what he has to tell us today.
Very good. So you see the main screen here. I can start?
Yes, you can start.
Yeah. Thank you for that kind introduction. Yes, it is a humbling and full of pleasure experience to speak at a symposium at Sharif University. I spent a good 4 years of my life walking around the hallways of the electrical engineering department and other parts of the university. And, it's a good number of years I haven't been there back in the university, although a few years back I came and I gave a talk in the math department. But it still has been a good number of years, and I can't wait for an opportunity to revisit and see, how the department and the university has changed. Anyway, so it is an honor. I'll tell you a little bit about the work we've been doing. I'll start by giving you kind of a broad overview of some of the ways we recently think about problems in my lab, and then dig into a very specific problem on the notion of timing in the brain and how this framework of computation through dynamics has helped us tackle that problem. So let's start. I guess the bigger overarching question in my lab is trying to understand the relationship between the brain and the behavior. How is it that the brain supports various types of behavior? And we ask all of our questions in the lab from the starting point of behavior. We're really, we're never sort of interested in how a particular part of the brain works. It's always a question of how a certain behavior comes about. And of course, down the path of answering that question is mechanisms and processes in the brain. Now, when we think about behavior, really, the behavior is best thought of in terms of a computation, right? So you have a certain inputs. For example, a simple behavior might be a decision making. You have a certain set of inputs that come to the system. The system is as internal states, and it has to generate certain types of outputs. I don't want to advocate an input-output sort of metaphor for the behavior, because it's not like that. But at the end of the day, behavior can be characterized in terms of relationship between a set of variables that are environmental, variables that are internal to the state of the body in the brain, and variables that are outputs of the system, how you move your hand and eyes and the rest of it. So really what we want to try to understand when we talk about behavior are the computational framework, are sort of the mathematical descriptions that explain how those variables are related to one another. Now, on the other hand, when we think about the brain, we have these groups of neurons that are in the brain, communicating with one another with various architectures, and they generate patterns of activity, dynamic patterns of activity. And one of the challenges in trying to understand the relationship between the brain and behavior is really the challenge of these two operate on two different currencies. When we think about the behavior, we think about those sort of this mathematics that are the computations that relate to the behavioral variables. And when we think about the brain, then we have to contend with the fact that we are dealing with variables and dynamic patterns of activity that are generated by the neurons. So in our hands, really the heart of the question of how to relate the brain to behavior turns out to be a question about how do dynamic patterns of activity perform computations. If we try to understand, if we manage to understand, or at least begin to understand how dynamics give rise to computations, behaviorally relevant computations, that's a proxy for trying to understand how the brain give rise to behavior. Now, in my lab, we're mostly interested in sort of more higher level, so-called cognitive computations. And those are fairly difficult to study because cognitive computations are those set of computations that are not immediately observable. You know, thinking, imagining, making inferences, planning, all of these are cognitive computations. And because they're cognitive, that means that there are a sort of set of unobservable or so-called latent variables that drive those computations. And we have to sort of figure out those latent variables from the analysis of the behavior and also look for sort of internal analogs in the dynamics of the brain. Now, what is really cognitive computations? It's hard to define. Even psychologists don't have a very good way of saying what's cognitive. I'll sort of introduce it with a very simple but fairly general formalism. I'll say, cognition are sort of those types of computations that we do that rely on abstract knowledge. The world gives us data, but the data that it gives us is oftentimes ambiguous, uncertain, and sparse. But we learn from that data structure. We learn abstract knowledge about how things operate, how things relate, how the things give rise to other things. And then we use that knowledge to make inferences when we're faced with new sparse data. If you know how a face looks like, then a partial face is all you need to figure out it's a face. So those are the inferences we make after having formulated the abstract knowledge about the world. Now, when we want to try to understand how the sort of these abstract information is formed and used, one of the fundamental things that we have to think about is sort of like, what is the nature of the underlying information? These are data structures, basically, that we have to deal with, data structures that are out there in the world, and then in the mind, they have to be emulated in that way. But to have a data structure that is appropriate, if you go with the computer science analogy, you need data structures that have the suitable form. Not all data structures are useful for all sorts of computations. And then when you think about the data structures that the brain has to use, you also have to think about how do you perform algorithms and intervene on those data structures. In fact, the entire limitations on what data structure is a good data structure is governed by whether I can run some interesting algorithms and interventions on that. And if I can, then that's a good data structure. So I can't remove this hide, but that's okay. I apologize for this being in front of it, but it's not a big deal. We will not have a lot of things at the bottom of that screen. So when we think about, for example, inferences that we make or data structures, the types of data that are out there in the world are of many different kinds. We have statistical sort of regularities in the world. There are relationships between things in the world. There are causal relationships where X leads to Y, and so on and so forth. And for each of these, we might have a different type of data structure that would make sense to have. For example, for statistical reasoning, we might have, probability distributions, prior beliefs, likelihood, evidence in terms of, probabilities, and have to combine them. And one formalism that people have developed to try to understand statistical reasoning is in the form of Bayesian integration, where you have these sort of probabilistic information about the state of the world, both based on your prior beliefs and based on the sensory evidence, and you have to combine them. So you need certain data structures and certain algorithms to be able to handle statistical information. When you think about relational reasoning, well, the world is now offers relational structures, But relational reasoning needs a different sort of a representation. You need to have, for example, hierarchical trees, a family tree, like, someone's the son of somebody else, someone's the daughter, someone's the uncle, someone's the father, and all of that. And that's a relational structure. You know that one, square in the city is closer to another one, but the other one is farther from another place. And those relational structures need a different sort of form of representation if you want to be able to perform flexible computations on it. Then you think about causal reasoning, you need yet another type of data structure. The data structure you might need sort of, like directed graphs. And all of these will require both a different structure to be embedded in the neural system and a different sort of set of dynamics so you can perform computations. Because if you want to, make a statistical inference, a relational inference, a causal inference, you need to run the dynamics on these different structures to be able to actually perform an interesting computation. And these these abstractions, at the end of the day, when you go to the brain side, are embedded either in the patterns of activity that neurons generate, which I call here activity space or A space. Here, what I'm plotting sort of a schematic of a pattern of activity that is generated in the state space of neural activity. Or it has to be embedded in the connections between the neurons, or weight space, which I'm pulling here as W space, right? So the synaptic weight between the neurons set the sort of tone of what the circuit can do, and then the circuit can generate certain patterns of activity by having that synaptic weights in it. So the question really here is that to generate abstract knowledge and the appropriate data structures, we should be thinking about what are the constraints on the weight space and the activity space that are suitable for a specific type of data structure and a specific type of computation. And when we think about sort of this in the context of the brain, now, what are the structures that exist in the synaptic weight space? Well, we have cortical modules, we have cortical microcircuits that are sort of specific architectures of connections. We have a panoply of circuits, certain group of neurons communicate with certain other group of neurons, but not other types. And sort of those are more anatomical architectural constraints in the synaptic space. And when you think about the activity space, then what you put in the synaptic space allows for the neurons in the activity space to have certain types of activity, that exist within some constrained manifolds, the representations that are either factorized, they're linearly decodable. Depending on the form of the representation and the synaptic structure, the form of the activity and dynamics that is generated in across the neurons will change. And all of this ties together. So there's a big puzzle here, is how do you go from behavior and computation all the way to understanding how is it that the weight space and the activity space are organized to embed the appropriate data structure and provide the means for performing algorithms and interventions on those data structures. And that's sort of what I meant by sort of computation through dynamics as a framework, thinking about computations on the brain side, dynamics and specific patterns of data structures that are embedded in neural space on the brain side to be able to connect the two. Now, so embeddings for the structure and dynamics for intervention. Those are the types of things that we expect to be finding in the neural space. Okay, so with that in mind, then I want to just tell you about a very specific example of this approach in our lab for studying the notion of statistical inference, which we have done so in the context of interval timing. So what I will be telling you is a project that involves several components, involves training and behavioral studies in non-human primates, where we, ask them to do certain tasks, and those certain tasks involve sort of taking advantage of statistical regularities in the environment, and also to perform in the context of a timing task. Then I will telling you about electrophysiology, and which is sort of a bread and butter of the work in my lab, which is to study neural activity at the level of single neurons and population to try to understand how then the dynamic patterns of activity could be mapped onto the latent variables associated with the behavior. And as part of this, we always are trying to form and validate or reject hypotheses by building specific computational models. These computational models serve as sort of foundations of our hypotheses. And within the context of these models, we operate to try to understand whether a hypothesis is validated by brain data or not. So we build these models and then we look at the interior of them, compare them to the brain, and we make inferences about the relationship. So before I get started, can I get a yay, nay from somebody so I know I'm connected, because I only can see my slide.
Yes, yes, you're connected. We hear you.
Okay, great. So let's move on. So let's talk for a moment about what is statistical inference. So think about a very simple situation where we have an agent that wants to act upon the world. So they see a stimulus here noted S, and they want to generate the action A. Now, the stimulus comes from the world, and the agent needs to operate on that stimulus, but what the agent had is not the stimulus, but a noisy measurement of that stimulus, because we have our sensory system that has, all sorts of biological noise on it. So what the agent has to do is to rely on a noisy measurement. And therefore, their actions will have some variability or some inaccuracy. Now, it turns out stimuli in the world oftentimes are governed by some statistical regularities. And if the agent were to learn those statistical regularities and have some prior expectations of what the stimuli in the world ought to look like, then the agent could combine its knowledge about the prior statistics with its noisy measurements and generate types of actions that are more accurate, that can perform better. And that's sort of the crux of Bayesian integration, or statistical inference more generally, which is to bring together various sources of statistical knowledge that we have about a noisy observation to generate better responses. Now, I usually, if I had the pleasure of being in your presence, we would do a few trials together for a specific task that I want to introduce to you, which we have used over the years to study statistical inference. We can't quite do that together, but I still want to show you a few trials of the specific experimental paradigm that we have used in my lab. So the specific experimental paradigm is a time interval reproduction task, or what we call a ready, set, go task. The task is very simple. An observer sees two flashes, which are the first two beats of a rhythm, like flash, flash, right? And then the observer's task is to generate the third beat of the rhythm that is omitted. So they don't hear the third beat, they only hear the first two beats, and they have to generate the third beat. So what they have to do is to measure the time between the first two beats and then basically reproduce that time, right? Now, I'll run a few trials for you. What you will see is a flash at the time of ready, a flash at the time of set. The flash at the time of the ready is the first beat. The flash at the time of set is the second beat. And although we can't do this together, I'd like to invite you to, for a moment, try to reproduce the third flash in your mind, or by clapping, or by internally generating the third beat. So you will see flash, flash, and then in your mind, I want you to clap at the time of the third beat, at the time of the expected third beat. So we'll do 4 trials together, and I want to demonstrate a very interesting property of this task. So here we go. You'll see flash, flash, and you generate internally the third flash in your mind. So here's the first trial. Let's do 3 more trials. Trial number 2. Trial number 3. And last trial. So usually when I demonstrate this in person, what happens is that people are quite unprepared for the 4th trial, because it's suddenly much longer than the first 3 trials. And this is a common feature. When we are uncertain about the measurements we make, we usually very quickly generate expectations based on previous trials. So all the first 3 trials were relatively fast, and people generate a quick expectation that it must be fast, but then the 4th trial turned out to be a slower one, and people kind of a little bit startled by that and make a lot of error. And that's sort of a symptomatic of a brain that very quickly generates expectations and uses expectations to improve its performance. Now, when we actually, ask human subjects to perform this task, they do exactly the same property comes from their behavior. So we give them sort of the sample interval from between the ready and set, and we measure the time they produce, TP, between the set and go. And if I were to plot for you the produced interval of the subject as a function of the sample interval, what happens is that although the produced interval generally increase with the sample interval, they exhibit these biases away from the diagonal, as you see here by the red arrows. They oftentimes overestimate the shortest interval and underestimate the longest interval. And the reason they do this is that by doing that, they sort of bias themselves towards the most expected range of intervals, which is the intermediate range. And although it may appear that these biases are actually costly and harmful, they're not. By generating these biases, people in fact do better. And why is that? Although bias generally is not a good thing and it increases error, it turns out in this case, by introducing this bias, subject simultaneously reduce the variance associated with these two endpoints. So the bias actually, the error generated by the bias is overcompensated by the reduction in the variance. And it turns out that the total error, the root mean square error in this situation, in fact drops when your behavior has biases associated with it. So this bias is sort of a way of optimizing the behavior in the context of this task by using knowledge about the prior distributions. And this is what humans do and also monkeys do. Okay, so we wanted to study this task in monkeys and try to understand how, in fact, these biases come about. But before doing that, we had a problem. And that was that we already actually didn't know, even the Bayesian integration and measurement aside, we actually didn't know how the brain produced time to begin with. Sort of the question of, if I were to ask you just clap after 1 second, how does the brain solve that problem, regardless of, measurements and distributions and so on and so forth. So I quickly will tell you how we tried to tackle this problem first and give you a quick summary of the answer to this, which I will need to be able to then tell you how the brain does the statistical inference in the context of a prior distribution. So first thing we did was that we actually trained monkeys to perform a simple time production task. There was no uncertainty. The animals had to fixate a central spot, and the central spot was either blue or red, and that indicated to the animal that either it had to produce a 1500 millisecond interval or an 800 millisecond interval. And the entire logic was to try to understand how does the neurons in the brain control the dynamics such that the animal can produce these two different intervals after the set. Okay, so this is the sort of a different task that we call the Q set go task. So to perform this sort of experiment, we focused on a number of brain regions. I'm focusing here on one of them, the dorsomedial frontal cortex. We actually recorded in dorsomedial frontal cortex, which is highlighted here as a sort of frontal region of the brain near the arcuate sulcus. We also did recording in thalamic regions that project to this area and the parts of the basal ganglia that receive input from the dorsomedial frontal cortex. But for today, I'm just going to focus on our recording in this region, the dorsomedial frontal cortex, which comprised of supplementary eye field, dorsal region of the supplementary motor area, and pre-supplementary motor area. So the first thing we did is that we actually inactivated this area in this Q-set-go task to make sure it's causally involved, and we verified that the inactivation of this area caused animals to become more variable than the timing task, even though they had no problem providing responses. And in this case, the responses were either a button press or making an eye movement to a target. So they had no trouble moving their eyes or hands, but their timing got impacted when we inactivated, which suggests that the DMFC is a node that is involved in timing. And of course, this was built on a large body of work that I have cited on the right. Many people had already suggested that the dorsomedial frontal cortex is implicated in interval timings, and that's actually how we chose this particular area to record from. Okay, so here after we verified that this area is involved, we went in and we started recording from individual neurons in this area. And here I'm plotting for you the firing rate of 6 sampled neurons. What I'm plotting for you is the firing rate of the neuron as a function of time. And it's aligned to the time of set. And you see a sort of a different shades of red. Those are when the animal was instructed to produce 800 milliseconds. And of course, animals have some variability. Sometimes they produce a little bit more, like 860, sometimes a little bit less, 740. And the firing rates were different in those cases. But those are the red shades. And all the shades of blue correspond to when the animal was asked to produce a 1500 milliseconds. And again, you see a sort of a pattern of activity that is different and can vary depending on what interval the animal actually produced. Now, when we recorded from these neurons, when we sort of started this work, we went in with a certain set of hypotheses that already had been stated before our recording. There were theories about maybe perhaps the brain produces time by a clocking mechanism, a clock and accumulator. So there will be clocks, and then there will be sort of a ramping activity that will accumulate the clock up to some threshold, and that's how people produce time. There were theories about, maybe it's involved in sort of oscillations, and the beat frequencies of multiple oscillators allows the animal to produce specific times. There were also other theories. What we found was not consistent with any of the existing theories out there. When you look at the neurons, we found the highly heterogeneous patterns of activity across the neurons. They couldn't be viewed as oscillations, all of them. They couldn't at all be viewed as sort of ramping activity or a clocking mechanism, but rather kind of complicated heterogeneous dynamics, just as you would expect from a blind network of neurons interacting with one another. But what caught our eye and was a very, very prominent feature in the data was what you can see and sort of validate here with your own eyes, which is it seemed like although neurons have highly sort of idiosyncratic firing patterns, there is something interesting about it seems like they stretch their response with the same general profile when they produce a longer interval compared to the shorter interval. So the profile of the response of a neuron don't seem to change much, but it seems to be stretched in time or compressed in time, depending on whether the animal produces a shorter interval or a longer interval. Not perfectly stretched, but that's a very, very strong feature of the data that we found across 70+% of the neurons. And so that's sort of a feature what we call temporal scaling, right? So the scaling of the neurons in time seem to be sort of a proxy of the control mechanism that the brain seemed to be using to produce different time intervals. Now, because these neurons were highly heterogeneous, it's kind of make it difficult to make inferences by looking at one neuron at a time. There's not a whole lot to be learned because they're so different. So there was another way to think about this. This is by looking at the state space. So, I don't have the state space analysis here, unfortunately, but it's important for me. So I guess I'll step back. Hopefully later will come. So I guess this slide is just to let you know that this idea of temporal scaling seemed to be the best hypothesis explaining the neural data. We compared it to all sorts of theories and models that were out there, and this by far was the best model in terms of explaining the neural data. Okay, yeah, here is what I wanted to tell you. Because the neurons are so heterogeneous, one way to think about them is look at more but collect in what is called the state space. So what is state space? State space is basically a high-dimensional coordinate system where each axis corresponds to the firing rate of the neurons. So if you have, 10,000 neurons, it's a 10-dimensional space where at each point in time, the firing rate of each of the neurons is the number of axes, okay? So now as these neurons change their firing rate throughout time, the point in that state will that's one way to sort of collect the activity of the neurons. Of course, we can't visualize a 10,000 dimensional space, but we can just at least conceptualize what it means to have a state space in dimensions. Now, what I would like to invite you to do for a moment is to think how does temporal scaling manifest itself in the state space representation? If I have a group of neurons that are changing their firing rate in a certain way, that will generate some trajectory. Now, if all the neurons get stretched by a factor of 2, what would happen in the state space? How would the neural trajectory change in the state space? I hope with some thinking you can verify for yourself that because stretching simply means that the neurons are getting faster or slower, in the state space, actually nothing will change. If you had perfect stretching of a factor of 2, then the neural trajectory will remain exactly the same, with the only difference being is that you would go on that neural trajectory with half the speed. Because effectively stretching is like changing the speed. So that means that in fact, representation in the state space has this cool feature that in fact, if the temporal scaling were a meaningful feature of the neural data, we would expect some invariance of the trajectory in the state space. Some part of the trajectory should not change at all if the temporal scaling was the main feature. And when we did this analysis, we found that indeed that was the case. The neural trajectories associated with the different intervals were self-similar in the sense that they all looked the same, but they just had slightly different initial conditions depending on the interval that the animal was going to produce at the end of the set go. So it seems like the neurons start from a different set of initial conditions, but then the neural trajectory evolving off of that initial conditions was quite similar. And of course, I have plotted this for you. As I said, there are like 700-800 neurons that we recorded from. Here, I'm plotting for you all just to visualize the neural activity on the three principal components of the neural data. Mind you that the actual neural data was fairly low dimensional, so we could explain a large part of the variance with just a few principal components. But the key feature here that I want to highlight for you is that really it seems like the timing, which was the original question we wanted to answer, gets controlled in the brain by setting an initial condition and then adjusting a speed. So the speed is adjusted. If the monkey is asked to produce a long interval, then the initial condition is adjusted such that the activity will flow slowly in the state space. And if the animal is asked to produce a short interval, the initial condition is set such that the activity evolves with a faster speed. Okay, so it seems like the key component is that the instruction, or what the fixation cue says, sets the initial condition, and that initial condition then sets the speed. So we can sort of schematically or algorithmically think about this as a very simple framework for how the brain produces time. Basically, the argument is that there is a instruction set the initial condition set the speed, and then the activity moves with a certain speed up to some threshold, and when it reaches the threshold, the animal produces either an eye movement or a hand movement. That will be a fairly straightforward control mechanism for producing time, okay? Now, having had this as a hypothesis for how the brain produces time, we can go back to our original question of the ready, set, go task, where the animal has to first make a measurement of that interval and then produce it. So the key difference between this task and the previous task is that the animal doesn't know at the very beginning what interval it has to produce. When the ready comes, it has to measure until the time of set. And then when the set comes, now the animal knows which interval it has to produce, and therefore it can produce it. Okay, so let's imagine what the result could look like, okay? So let's say we're exposing the animal to 5 different intervals shown here by different shades of gray. If our idea of initial condition is correct, those 5 shades of gray should go to 5 different initial conditions, and those 5 initial conditions should lead to 5 different speeds, okay? So first, let's think about what should happen if there was no Bayesian integration and there was no bias. Well, if there was no Bayesian integration and no bias, then as I've plotted in the right, we expect the produced interval to be perfectly aligned with the sample intervals, and therefore we expect the initial conditions to be equidistant because the samples are equidistant, right? But we know that this is not how the animals perform this task, or humans. We know that the responses are actually biased. That is, the responses that the animal gives to the shortest interval is biased towards the middle, and the responses to the longest interval is also similarly biased towards the middle. So what does that mean for our theory of initial conditions and speed? Well, what it means is that those same biases should present themselves in the initial conditions. The speed needs to be biased, and therefore the initial conditions needs to be appropriately warped. Especially the two ends of the initial conditions need to come closer to the middle points, because that's effectively the regression to the mean, right? So the question here is, how does the brain measure the interval between the ready and set such that these initial conditions are appropriately warped? Sort of this is the question of effectively the statistical inference, the Bayesian integration. The Bayesian integration has to happen here, where you make a measurement, but you have a prior, and based on that prior, the measurement has to be appropriately warped so that the initial conditions are regressing towards the mean, right? Okay. So it turns out, I'll tell you a little bit about the details, but it turns out that, you know, the answer is a very cool result that the cool solution that the brain has found, which is it measures the time during the ready set through a curved trajectory. And sort of a very interesting feature of a curvature is that if you project the points of a curvature onto a line, the projections become warped, just as I have plotted here for you. So how did we come up with this sort of hypothesis? Before even recording, we have become in my lab accustomed to try, when we don't know how the brain solves a problem, what we often do is that we go to some modeling effort and try to use network models to gain some insight of how the brain could solve the problem. So in this case, we trained some recurrent neural network to solve the task. We gave it sort of the ready and set, and we asked it to produce the time. And when we gave it noisy inputs, some of these networks learned to solve the task with the property of Bayesian integration, with a regression to the mean. And after we found some networks that were basically replicating the behavior of the animals, we could then open them up and ask, what is the internal representations in these networks? And what we found was indeed that in the internal representation of the networks, the representations was curved at the domain of the prior distribution, in this case between 800 and 1200 milliseconds. So there was a curvature that was generated by, in the neural trajectories during the ready and set, associated with the domain of the prior. And we sort of set out as, okay, that's an interesting hypothesis. And of course, it's an interesting hypothesis for Bayesian integration, not just because it's curved, but also because it also explains the drop in the variance, right? So here, sort of schematic, if I were to draw for you a little curvature here with time, if we were to project the points off of the curvature onto that line, not only the points will be warped, as we would expect from the initial conditions, but also variances, which here are shown by this sort of shaded ellipses, will become shrunk near the edges, right? Near the edges, that variance along the trajectory gets shrunk to a much smaller value. And that's exactly what a Bayesian integrator would do, a Bayesian observer would do. It will create the biases that will appropriately reduce the variance, right? So this seems like a reasonable hypothesis for how the brain might solve the problem, and that's also what we found in our networks anyway. Okay, so we set out to test a very specific hypothesis, which I call the curvature hypothesis. The hypothesis was that if we were to expose the animals to perform this task with a prior distribution, we ought to find a curved trajectory during the ready set, sort of tuned to the domain of the prior. And then when the set comes, the activity has to be projected onto some line such that the initial conditions are warped. And those warpings should lead to biased speeds. And the biased speeds then should lead to a biased behavioral response, right? That's a full-blown explanation of the behavior based on this hypothesis. So to test this hypothesis, we trained monkeys to do a task just like the one I showed you, the ready, set, go. The monkeys would fixate a central spot and hold their hand on a joystick. After some delay, a flash around the fixation will present the ready. Then after the sample interval, another flash will represent the set. And the monkey's task after the set was to reproduce that interval, the TS, by either a eye movement to a target that was presented throughout the trial, or by deflecting the joystick at the appropriate time, at the time of the third beat. Okay? So we trained monkeys to do this, and we trained the monkeys to do this with two prior distributions. That is, the sample intervals that the animals were faced with were either sampled from what I call the short prior distribution that range between 480 and 800 milliseconds, or with a long prior distribution that varied between 800 and 1200 milliseconds. Okay, so we trained monkeys with these two priors, and we did these two priors because by having two priors, we're in sort of this advantageous situation where we can explicitly look at the effect of the prior, as I will discuss a little bit later. Okay, so, and the animals were rewarded for producing the time perfectly. That is, the animal were not rewarded for being biased. The animal had to produce the interval as perfectly as possible, but of course, to optimize for that, they had to generate the bias. Okay, so here is the behavior of one animal on one typical session. I'm plotting for you the produced interval on the y-axis, TP, as a function of the sample interval on the x-axis, TS. In red and blue shades are the two prior conditions. What you can appreciate, the small dots are individual trials and large open circles are the averages. What you can appreciate is that generally the produced intervals are increasing almost linearly with the sample intervals. But both for the short interval and the long interval, there are these sort of biases away from the diagonal towards the sort of the respective mean of the two priors. And one sort of very telling feature is that if you focus on the 800 millisecond interval, which is the overlap between the two prior distributions, what you find is that the animal is now producing the exact same interval, 800, for the two conditions, such that they're underestimating that 800 when it's coming from the short prior, and they're overestimating it when it's coming from the long prior, right? And that's a very strong evidence that the animals are relying on the prior distribution. And one thing I forgot to tell you is that the task, what we do is that the color of the fixation on every trial tells the animal which distribution the interval is sampled from. When the color of the fixation is red, the animal expects the interval to come from the short prior. And when the color of the fixation is blue, the animal expects the interval to be from the long prior. And you can see there is a circle and a square on the screen. That's just to tell the animal whether to respond with the eye or the hand. So there are two sort of effector modalities, hand and eye, and there are two different priors, and all of that is cued at the beginning of every trial, and it's randomly changing from trial to trial. Okay, so what you can see is that the animals kind of on a trial by trial can switch between the two priors, and the responses are appropriately biased towards the mean of that prior. That's an indication that the animal is actively integrating the prior distribution. Okay, so let's go to the neural data. First, I want to plot for you the activity during the set-go, which is the part that we had already studied in the Q-set-go task, right? So in the Q-set-go task, we found that what the neurons do is that they go to different initial conditions, and then they generate dynamic patterns of activity with different speeds up to some threshold. Here is the activity of sort of 6 individual neurons that we've recorded in different conditions, hand and eye, in the 2 monkeys. And you might see certain features of temporal scaling, which become heavily evident if I were to plot for you in the state space the neural trajectories. So what we found is that when the set is presented to the animal, the neural activity suddenly gets perturbed and goes to some initial condition, which here is marked at sort of set plus 200 milliseconds. And from that point onwards, the neurons the patterns of activity evolved with different speeds. And you can appreciate these different speeds by these dots that I have put on the neural trajectories. So the time between every neighboring dot is 20 milliseconds. And what you find is that for the red trajectory, which is the shortest trajectory, the distance between the dots is much wider than for the light blue. And that's because the light blue trajectory is moving slower. So in other words, during the set go, we didn't find anything different from what we had found in the Q set go. Basically, the principle that we had found in the Q set go directly generalized to the ready set go, in the sense that the animal was using, so the brain was using some sort of an initial condition that set the speed, and that speed then determined the interval. What about the ready set? Well, in the ready set, we found neurons that were generating very, very different patterns of activity depending on the two priors, and highly heterogeneous. It was not necessarily all ramping up or, oscillation or things of that kind. But when we looked at the neural trajectories, what was a striking finding was that indeed the neural activity established these curvatures that were tuned to the domain of the prior. So the curvature for the short prior was sort of started around 480 and ended at 800, and for the long prior for about 800 to 1200. Sort of there were two self-similar curvatures associated with those priors for the duration of the ready set, which was consistent with the idea that perhaps the curvature is a trick that the brain is using to generate initial conditions that are appropriately biased. Okay, so this sort of on the surface seems consistent with the the curvature hypothesis. So we set out to actually test that hypothesis rigorously and quantitatively, every step of the hypothesis, from the curvature to the initial condition to the speed to the behavior. So the first question was, are the curvatures consistent with Bayesian integration? That is, if I were to project the points on the curvature onto a line connecting the shortest to the longest interval, would the biases match the biases that we see in the animal's behavior? And what I'm plotting for you here is exactly that. So I'm plotting for you that the produced interval as a function of sample interval. The dots are actually projections of the neural data during the neural trajectory, and the lines are the Bayesian fit to the animal's behavior. And we find a remarkable match between the biases expected from the found in the behavior and expected from the projections of the curved neural trajectory, suggesting that indeed these curvatures could be a substrate for generating the appropriate biases. Here I'm plotting for you one of the conditions, one of the monkey's hand response, but for all the conditions, this behavior was replicated. And another thing that we did is that we also looked at the projection of the variance of the projected points. And indeed, what we found was that across all the conditions, the variance had this sort of inverted U shape, meaning that the variance was variance of the projected points was smallest at the two edges of the curvature and largest in the middle, which you would expect again from the bias variance trade-off of Bayesian integration. Next, we asked the question that is it the case that then the initial conditions that are warped are related to the speed? And what we found was indeed the speed of the neural trajectories was increasingly slower for projections that were farther away or closer to the longer interval. So indeed, the initial conditions was again correlated strongly with the speed of the neural trajectory. And more impressively, what we found was that the initial conditions were predictive of not just the speed associated with the true sample interval, but were associated with the biased sample interval. For example, if you look at the top right plot, what we found was that the initial speed right after the time of set, okay, was lower than what would have been expected from an unbiased profile. And when, of course, the speed is lower, that means that the interval that the animal will produce will be longer, which is exactly what you expect. For the shortest interval, you expect the animal to produce a longer interval, so you expect the speed to be lower at the lower level, and that's what we find in these neural trajectories. So in the neural trajectory, speed of the neural trajectories were consistent with the Bayesian estimates and not with a true sample interval, which is consistent with the animal's behavior. And finally, the speed of these neural trajectories were predictive of the interval that the animal produced. So as a last step, it seemed like the speeds could explain the animal's behavior. So all in all, we had a pretty nice sort of a match between the neural data and our hypothesis, which consists of, the prior leading to a curvature, leading to warped initial conditions, leading to biased speeds, and finally leading to regression to the mean behavior, okay? So stepping back for a moment, what have we learned about statistical inference through this sort of computation through dynamics framework? Well, in sort of stepping back from timing, I want to tell you what seemed to be sort of a principled way of thinking about this. So imagine that the brain wanted to make an inference about a stimulus S, okay? And the stimulus S has some prior distribution P of S, okay? So the P of S here is shown as some Gaussian where the two ends are low probability and the middle of it is high probability. So a Bayesian integrator wants to bias itself towards the middle, right? And the question is, how does the brain do that? And what have we learned from this sort of effort in the timing? Well, what we have learned is that although time is a scalar variable, the brain seemed to be representing the time in this task through a curvature, which is two-dimensional, not a single dimension. Although time could be represented in one dimension, the brain is representing it as two-dimensional. So what it's doing is that it's, instead of representing that stimulus S in one dimension, it's sort of exploiting one extra embedding dimension to represent that interval, that stimulus in. Now, what does happen when you take advantage of an extra embedding dimension? Well, if you represent a scalar variable in two dimensions by adding an embedding dimension, then if you were to decode the, oops, I apologize. Sorry, I have dogs that bark. Okay. So what happens with the extra embedding dimension is that it allows you that if you were to now read out this representation on a line, now these representations will be warped, right? The points in the ends of the representation get squished together, and the points in the middle are, relatively speaking, get a more representational space. And this warping is exactly what you want to be have a biased representation towards the mean. And now if you make a measurement that is noisy, the inference will automatically be biased towards the parts that have a larger representational space. And I want to, for those of you who are familiar with this is actually fundamentally the notion of efficient coding, right? You give a larger representational space to the parts of the world that are more probable, which is exactly efficient coding. And that actually links it to a lot of work that has been done in early sensory areas in the visual system and auditory system, where people have proposed that the way the brain incorporates natural statistics of the world is by efficient coding. It is by providing more representational space to the parts of the world or to features of the world that are more probable. And what we find here is that in a much more sort of short-term learned framework and in a completely different part of the brain, in sort of prefrontal premotor areas, the same principle presents itself but through dynamics, through the dynamics of neural trajectories. So for statistical inference, it seems like the data structure that the brain is relying on is sort of warped representation, get the initial representation and appropriately warp it such that the prior is now embedded, okay? And then read off of that warped representation to be able to get the appropriate estimate, okay? Since we're at the end of the time, I want to tell you a little bit about something else, but since we're at the end of the time, I'll wrap up here. We have also learned something about what is really happening during the measurement. I guess very quickly I'll tell you, during the production, we have found the speed. And during the measurement, I showed you these neural trajectories, but some additional finding that through this computation through dynamics we have learned is that if I showed you these two neural trajectories from one perspective, but if I rotate them on their head, what you find is that there actually are parallel pathways, right? And interestingly, the speed of these two is different. So when the animal is measuring interval in the short prior, the neural trajectories in the ready set now are faster than when the animal is measuring the interval during the long prior. And it turns out what the brain is doing, this is a more recent work that is also published, but what we found is that in fact what the brain is doing is that already during the measurement, the brain is adjusting the speed of the dynamics such that it expects to reach a certain point in the state space at the expected mean of the distribution. And of course, the short and the long have different means, which means you need two different speeds to reach that threshold at the appropriate time. And the way the brain, in fact, measures time is not by measuring absolute time, but the brain measures times relative to the expectation by adjusting these speeds. And it turns out not only this is true for the two prior, it's also true when we subject the animal to a completely new distribution during learning. So during learning also, these neural activities change their speed gradually such that the activity reaches the appropriate threshold at the appropriate new mean of the distribution. So if you move the distribution, the speed slowly changes such that it reaches the threshold at the position of the new mean. Okay, so what we have learned about timing through this sort of computational through dynamics are two things. At the time production, we found sort of self-similar low-dimensional dynamics, and the speed of the neural trajectories, which is fundamental to studying dynamics, encodes the desired duration. And for measurements of time intervals, again, we find self-similar curve trajectories, and the speed encodes the expected duration, although the actual duration might be a little bit longer or shorter. And that sort of has led to this general framework where you, at least for the timing, you can think about the timing as a computation that happens within the recurrent system of the neural machinery that connects the frontal cortex to various other parts that I didn't have time to talk about. And the way the timing goes about is that there is some contextual input that is the prior, and that helps set the appropriate set of curvatures and warpings in the system. And then there is some timing information that either can come from an instruction like the cue set go, or from memory or measurement in the ready set go, or from other cues that we haven't discussed. And that could be a sort of a machinery that does a generalized timing and statistical inference. Okay, with that, I think we're at the end of the time. Let me thank the people who actually did all the work. Jing Wang and Egbal and Devika Narain did the early work in the QSET Go, where they developed, Devika developed the models, and Jing was sort of the main force for the physiology, and Egbal helped in trying to figure out how the brain produced time. And then on top of that, the work that my current, Jing and Devika both are now have left my lab, and they're running their own lab. Egbal is a new graduate student at MIT in a different lab. Then my current senior postdoc, Handsome, did the work, most of the work on the Ready, Set, Go first part on the discovery of the curvature with the help of Devika on the modeling side, and some data analysis with Nico. Nico is now also graduated and has left my lab, but he also was the one who spearheaded the learning experiment that I didn't have time to go to. And with that, I'll thank the funding agencies and thank you for your attention. I think I must be close to the end of time, right at the end of the time.
Thank you so much, Mehrdad, for the very interesting talk. So as we are very close to the end of the time for the talk, I've posted the panel session here, which people can go to soon to ask more questions and interact with you for the next 30 minutes. As I won't be able to join you in the panel, I have one question that I just wanted to clarify. So you mentioned something about reduction of variance for the data points that were far away from the mean. Now, but as you know, in Bayesian integration, the variance of the posterior only depends on the variance of the evidence and the prior. So it doesn't. So the variance normally doesn't depend on the mean of the evidence, unless you think that the evidences have different variances. Do you know, like in Wiener filtering or Kalman filtering, the sigma for the posterior only depends on the sigma for the evidence and for the prior, not on the mean. So I'm wondering, how would you explain the reduction? And why do you think first of all, from a normative perspective, there should be a reduction, and then you also observe it. But that's the observation. That's the data. But I just want to know, right?
Yeah, I understand the question. So there are a couple of topics here to tease apart. So there is a prior distribution that has a mean and variance, let's say. There's the likelihood function or the conditional measurement that has a mean and variance, right? And you combine them and you generate a posterior that has a mean and variance. None of this is related to the behavior yet. The behavior is the point that you pick off of the posterior distribution, right? So you pick one point off of the posterior distribution. For example, its peak or its mean, and that's your estimate. That estimate is a point estimate that you take off of the posterior. Now, across trials, that estimate also has a distribution.
But the distribution is not the same as the posterior? I thought that...
No, it's not the same as the posterior.
I see.
Even for two Gaussians, it's not. Even if the likelihood was Gaussian and the prior was Gaussian, the variance of the posterior and variance of the posterior mean are not the same.
Yeah, true. The variance of the mean... Yeah. So, I understand that. So, so the posterior has a variance, and that variance is only dependent on the variance of the prior and the likelihood function, right?
Yes, exactly.
Right. So you're talking about something like the standard error of the mean, something like that. So you mean like all the samples that you're talking about the sample mean variance, something like that. Is that
exactly? So prior, of course, is not changing across trials.
Right. Posterior
prior is the same.
Right.
What is changing across trials from trial 1 to trial 2 to trial 3 is the measurement, right? And each time you have a different measurement, that leads to a different posterior, and that leads to a different estimate. Okay. Now the estimates, if you combine the prior with the likelihood, the estimates all get biased, shrunk, close to the mean of the prior, right? And the variance of the across those distribution of estimates also gets shrunk because the means are shrunk. So if you get all your estimates and you push them towards the mean of the prior, the variance across them gets smaller. That's
across all the means, I agree. But you were saying just for the evidence that they're far from the mean, you were saying that the variance of those we have a biased measurement, but with reduced variance for that certain mean, and that's
exactly what a Bayesian does. So let me try to bring that. So if you actually forgetting about the neural data altogether, right, and just looking at a Bayesian observer, you apply a Bayesian observer to perform the ready, set, go task, right? What you will find is that their behavior will show this bias.
True. The bias is understandable. So always
and the variance of the estimate will be an inverted U curve.
Yeah,
that's just coming from a Bayesian observer. The reason for this is the following. So you see this prior here, which is a uniform.
Yes.
So if I imagine I plot a likelihood function over it, okay, a Gaussian over it.
Oh, maybe
if I put the
could it
So if I so if I put the Gaussian in the middle, because it's multiplied by something that is relatively uniform everywhere, it doesn't reduce the variance much.
Okay.
But if I put it at the edge, it squashes half of the distribution, so the variance drops.
So then the prediction is that if you use a Gaussian prior, you shouldn't see the reduction in variance. If your prior...
No, you still see it. You still see it. You still see the variance dropping, but the form of it will not be exactly the same. So if you, for example, I mean, this is like the extreme, a uniform prior is like a Gaussian with an infinite variance, right? So as you make the prior narrower and narrower and narrower, near the middle, you will have now more and more reduction of variance. But what the key is the relationship between the likelihood and the prior. So if the likelihood is very narrow, even for a Gaussian near the middle, you will have little drop. But this is all relative, right? So what you might, I guess, this inverted U might give the impression that at the middle there is no drop of variance. At the middle there is a small drop of variance, at the edges is a large drop of variance. Does that
Yeah.
make sense?
Yes, yes.
Because you always are taking part of the distribution out. So for Gaussian, the form will be different.
Yeah, yeah. I
Yeah.
guess for Gaussian, the form will be the same as, so when you have conditional, anyway, for jointly Gaussian distributions, we know the exact mathematical form. But I think
Yeah.
since you use the uniform prior, then it's totally understandable that you see this reduction of variance more in the edges, of course, than in the center. Thank you so much.
Exactly.
So
Of course.
yeah. So if I may kindly ask you and everyone else to please join the panel and continue this wonderful discussion with Mehrdad, and I'll thank you, Mehrdad, for accepting our
Of
invitation.
course.